Today, we are going to present the application of nanotechnology in pharmaceutical and medicine industry. This is our team. I'm Helena.
And I'm vaishu.
Alright, so in this video, we are going to cover 3 examples of nanotechnology in the medicine industry. So, for me, I'm going to cover the example of solid lipid nanoparticles in drug delivery system, which are called SLN.
And I'm going to cover about super paramagnetic ion oxide nanoparticles (SP ions) in MRI and silver nanoparticles (AGNBS) in antimicrobial treatments.
For the properties of SLN, solid lipid nanoparticles are tiny particles, usually from 50 to 1000 nanometers in size, used to deliver drugs. To ensure quality, SLNs are characterized by their size, surface charge, shape, and drug loading capacity. They have high drug encapsulation efficiency, providing controlled and sustained release of medications. Made from biodegradable and biocompatible lipids, SLNs are safe and well tolerated. SLNs are stable and protect drugs from degradation by light, oxygen, and enzymes. They can be modified to target specific areas in the body. Scalable production methods like high-pressure homogenization are used to make SLNs. Not only that, there are many ways to prepare SLNs. Firstly, by high-pressure homogenization (HPH). The lipids are melted and mixed with the drug. This mixture is added to hot water solution with a surfactant, a substance that helps to mix oil and water. This hot mixture is forced into a high-pressure device to break it down into tiny particles, forming nanoparticles. The next method is solvent evaporation. Basically, the lipid and drugs are dissolved in a liquid that is volatile, like alcohol. This solution is mixed with water that contains a surfactant to make an emulsion. This liquid is then let to evaporate, leaving behind tiny solid lipid particles with a drug inside. Next, we have the microemulsion based method, whereby the lipids, surfactants, and water are heated up to form a microemulsion, a clear and stable mixture. Once formed, a cold water is added quickly, causing the microemulsion to break into tiny solid particles. Now you guys might be wondering, why do we create SLNs? There are many reasons for SLN's creation. First of all is for cancer treatment. SLN helps these drugs target cancer cells better and reduce side effects. Not only that, for fungal infections as well. SLNs reduce the toxicity of these antifungal drugs and improve their effectiveness. Next are viral infections. SLN enhance the stability and delivery of these antiviral drugs for infections like herpes and HIV. How does SLNs work? Basically, solid lipid nanoparticles are tiny and smart carriers designed to deliver cancer drugs directly to tumor cells. These particles have a solid lipid core that contains the drug, surrounded by a stabilizing shell. SLNs also coated with the molecules that are specifically bind to cancer cells, ensuring precise targeting. Once inside the cancer cells, SLNs break down and release the drug slowly, improving the effectiveness and reducing the side effects on healthy tissues.
So here are the references that we use to refer for gaining the information about this nanoparticle. Done. Thanks, Helena. So now I'm going to continue about the second example, which is super paramagnetic ion oxide nanoparticles, (SP ion). So what are the properties of these SP ions? We have two properties of SP ions. The first one is ultra small super paramagnetic ion oxide nanoparticles, which is smaller than 50 nanometer in diameter, used for imaging similar anatomical structure or for lymph node imaging. The second one is super paramagnetic ion oxide, which is a larger particles typically used for liver and spleen imaging. So, do you guys wonder how do they create SP ions? So, SP ions are created by the method called core precipitation. This is a widely used method for synthesizing SP ions. Iron salts like FeCl2 and FeCl3 are core precipitated by adding a base like NaOH or NH4OH. So, the first step is we need to dissolve iron salt in water under an inert atmosphere to prevent oxidation. Secondly, we need to add a base to the solution to precipitate the iron oxide. Thirdly, we need to heat the mixture to promote crystal growth. And lastly, we need to wash and purify the nanoparticles by magnetic separation or centrifugal. So, why are espions created? Espions are created to leverage their unique magnetic properties for a wide range of application, particularly in enhancing MRI, improving drug delivery, enabling hyperthermal therapy, facilitating cell tracking, advancing biosensing technology, and addressing environmental challenges. Their development represents a significant advancement in nanotechnology and its application to solve complex problems in various fields. For example, enhanced MRI contrast agents in magnetic resonance imaging (MRI) is a non-invasive imaging technique widely used in medical diagnostics. Traditional contrast agents sometimes have limitations in terms of effectiveness and safety. SPIONs offer several advantages as MRI agents. For example, superior contrast. SPIONs enhance the contrast of MRI imaging, particularly in T2-weighted imaging, making it easier to distinguish between healthy and diseased tissues. The second one is targeting imaging. Functionalized espions can be designed to target specific cell or tissues, providing more precise imaging for early diagnosis and treatment monitoring, and many more. So how does the espions work? Espions are widely used as contrast agent in magnetic resonance imaging (MRI). Due to their strong magnetic properties, which enhance the contrast of images, they are particularly effective in T2-weighted imaging, where they create dark hypotenuse region in the image. Liver and spleen imaging. Espions are taken up by the RES, making them useful for imaging these organs. Tumor imaging can help in identifying and characterizing tumors due to their enhanced permeability and retention effect. Second is cell tracking. Labeling cells with espions allow for the monitoring of cells migration and distribution in vivo. The last one is liver and spleen imaging. Espions are taken up by the reticular system, which is RSS, for making them useful for imaging these organs. So here are the references that we use to find these espions information. Last but not least, our third example is silver nanoparticles. So what are the properties of the silver nanoparticles? Typically, silver nanoparticles are between 1 to 100 nanometer in size. They can be spherical, rod-shaped, or even triangular, affecting the interaction with microbes. What is the surface chemistry of this structure? The surface of AGNPS can be functionalized with various groups to improve their stability, biocompatibility, and targeting efficiency. AGNPS exhibits strong antimicrobial properties due to their ability to release silver ions, which is Ag+, which interact with microbial cell membranes and intercellular components. How does the silver nanoparticle created? Okay. Silver nanoparticles are often prepared using chemical reduction methods. Silver nitrate, AgNO3, is reduced to silver using reducing agent like sodium borohydride, NaBH4, in an aqueous solution. Stabilizers such as citrate or polyvinylpropionidine (PVP) are added to prevent agglomeration. Why do we have to use AGNPS in this medical industry? Well, AGNPS provide broad-spectrum antimicrobial activity against bacteria, viruses, and fungi. They can disrupt microbial cell membranes and interfere with their metabolic processes. The next one is silver nanoparticles can overcome microbial resistant mechanisms that affect traditional antibiotics, offering a solution against multi-drug resistant strains. Last one is reduced toxicity. When properly formulated, AGNPS allowed for lower doses of antimicrobial agents, minimizing potential toxicity to human cells. How does AGNPS work? Well, cell membrane distribution. AGNPS attached to microbial cell membrane, causing structural changes and increasing permeability, leading to cell disease. Reactive oxygen species (ROS) production. AGNPS generate ROS, which induce oxidative stress and damage cellular components like DNA, protein, lipids. Interaction with biomolecules. Silver ions released from AGNPS bind to microbial DNA proteins, distributing replication and enzyme function. Lastly, Biofilm penetration. AGNPS can penetrate and distribute biofilms, which are protective layers formed by microbial communities, making the microbes more susceptible to microbial agents. So here are the references that we use to find information about AGNPS.
Throughout the entire making of this video, these are our roles and responsibilities. That's all from us. Thank you.